Remember when Willy Wonka said, "Everything in this room is edible. Even I'm edible," but that would be called cannibalism. It's looked down upon in most societies. Well, that brings us to today's topic, the laughing death, or Kuru, a rare disease of the nervous system that impacted the Fore people of Papua New Guinea at epidemic levels during the 1950s and 60s. Although usually referred to as the laughing death, the name Kuru actually means shiver or trembling in fear in the Fore language. Poor coordination, muscle twitches and tremors, difficulty walking, difficulty swallowing, slurred speech, moodiness and behavioral changes, headaches, arm and leg pain, and random compulsive laughing and crying. These are the effects Kuru has on one's body. Horrifying, isn't it? Death usually occurs 6 to 12 months after symptoms begin, when individuals become unable to stand, eat, and function on their own. Another worrying aspect of this disease is the incubation period. The time between contraction of the disease to the start of symptoms is on average 10 to 13 years, though it can extend beyond 50 years, making transmission difficult to monitor. You might be wondering, what gave birth to all this? Well, let me give you a helping hand. This bizarre disease was caused by a funeral ritual of the Fore people that involved eating the brains of dead relatives. The practice was based on the belief that the dead being eaten by their loved ones was better than being eaten by worms and insects. Women and children were most affected by Kuru as they prepared the body for the feast. To make matters worse, mothers would feed their children bits of the feast as snacks. Next time your mom passes you a bag of Chex Mix, write her a thank you letter. In addition, women were the ones who consumed the brains of the dead, while men mostly ate other parts of the body, thus being less impacted by the disease. So how did all of this come to light? Well, biologists hypothesized that the disease was caused by a virus that was spread by eating the brains of the dead. To test this, the NIH conducted a study in which they injected infected human brains into chimpanzees and watched symptoms of Kuru develop in the animals months later. So we know that the disease is transmitted through consuming contaminated tissue, but how does this actually work? We now know that Kuru is caused by prions. There's a lot that we still don't know about prions, but we do know that they are the misshapen forms of normal proteins. In Kuru specifically, we're dealing with the PRP protein. While prions are not living organisms and do not reproduce, they convert correctly folded proteins of their same kind to their own misshapen form, thus destroying healthy proteins and creating even more prions. These growing collections of prions clump together and accumulate in brain tissue, killing healthy brain cells and creating holes in the brain. This process of prion-caused brain deterioration leads to various diseases under the umbrella term of transmissible spongiform encephalopathies, aka TSCs. Some TSCs affect humans, such as Kuru and Creutzfeldt-Jakob disease, and others affect animals, such as mad cow disease and scrapie, which affects sheep and goats. Because we don't yet know how or if it's even possible to destroy prions and stop their growth in the body, these diseases have no cure. Because the practice of cannibalism in New Guinea ceased in 1960, the disease has become progressively rarer. However, because of the long incubation period, now only older people who contracted the infectious agent decades ago continue to develop Kuru.